Okay, so this is a continuation of our cells conversation that we were having. We had started information on this chapter last time, and we had gotten through the first portion of it, and then we're going to come back and finish up the rest of the packet. So we've gotten through about the first 5 slides into the cell membrane and then the organelles, and we're picking up with the passive movement across the cell membrane. So that's where we're at in the packet. Before we get too far into that conversation, though, I do want to talk about a couple of different things. Weather has not been friendly to us this semester. This is our second reschedule because of weather issues. And I just want to make sure that everybody is aware of the fact that I am going to kind of restructure some things moving forward so that we still get all of our face-to-face time in, and that labs and things like that we're going to be able to incorporate in. I don't know exactly what that looks like yet. I don't want to rush into that and then have to make a bunch of changes to it, but I am going to make sure that by the end of the semester, you guys have all of that face-to-face time, all of the labs that any of my other classes would have covered in first semester, you guys get as well. So kind of bear with me for the next couple of days, and I will get that restructured and I will discuss that with you guys, what that's going to look like. Um, just as a reminder, we were going to meet today, February 15th, uh, in order to make up for us not being able to meet on the 22nd. So on your schedule, it says 22nd was supposed to be a face-to-face, but I have conferences that night, so we were going to meet today to make up for that. Then weather hit, and here we are. So we're not meeting face to face tonight, obviously. We are not going to meet on the 22nd. And by that time, I will have a schedule in place and we will know what we're going to do from that point on. So just bear with me and I will get that posted to all of you guys in the near future. So looking at the packet, we're moving into the concept of moving things across the cell membrane. And if you think about the cell and go back to the concept of your cells being tiny little factories, they're more than just building blocks. They're more than just structural. They are actually producing things 24 hours a day, 7 days a week. And in order to build material, you have to bring things into the cell so you have enough supplies to build things. And then once you have these products built, you have to ship them out so that they actually can be used at different places in the body. So the design of the cell membrane is key to that because the lipids, remember those lipids with the round little heads and the two tails, because they are not actually fastened or fixed to each other, it allows for things to be able to slide in between them, in and out of the cell. So when the cell needs more products or more supplies so it can build things, it will pull those into the cell and it just slips between the lipids. And once those things are built, then it can push them out the other way and send them out to be used in different places of the body. So the design of that cell membrane is important in so many different ways. Now, the key thing about passive movement is going to be the predominant way that things are moved in and out of your cells. And on the surface, that doesn't really sound like it's that important, but it's actually very critical to our survival. We are very complex organisms, and in order to do everything that we need to do, we are trillions of cells in size. And when you think about that, trillions of cells, things constantly moving in and out of your cells, if it took energy to do that, there is no way we could consume enough calories to keep that system working. We would actually have to be much less complex or we wouldn't be able to survive. And so the fact that a majority of things that move in and out of our cell are able to do that without needing energy to make it happen is crucial to our survival. So passive movement is a very important process because it allows us to have all the cells that we have to move things in and out when they need to be moved so that we can continue to function the way that we need to. So when we look at passive movement, there's a couple different examples. And if you look at the first one, it talks about diffusion. Diffusion is the movement of any type of molecule across the cell membrane, not needing energy, and moving in a direction from an area of high concentration to an area of low concentration. You guys see examples of this all the time. We just usually don't put the terminology of diffusion to it. So if you have an air freshener and you spray the air freshener in the air, it's highly concentrated in the can. When you spray it, it moves out into the air where there's not a lot of that scent or that smell. So it's moving from high concentration to low concentration. That's why if you spray air freshener in the back of the room, eventually those people in the front of the room are going to be able to smell it because those molecules are moving from where they're highly concentrated and then spreading out to areas where they're low concentration. You've ever spilled a glass of milk? You guys know that in the cup, high concentration of milk, and when you spill it, the milk doesn't stay in the cup. What does it do? It spreads out and it goes from where it's highly concentrated to where it's in low concentration. So examples are all around us of diffusion happening. Like I said, we just don't put the term diffusion to it. Now, when we talk about diffusion, I said it's the movement of molecules. And so when we're talking about this in reference to cells, it could be oxygen molecules, it could be sugar molecules, any small molecule that is moving in and out of the cell is going to use the process of diffusion. Now, I do indicate small molecules, and that's going to come up here in a little bit. The other type of movement that we have is facilitated diffusion. And when we use the term facilitated, that simply implies that there needs to be some sort of help in order to get this done. If you have a facilitator for a meeting, they're the ones that are going to be kind of overseeing it, make sure that things go the way that they're supposed to. So with facilitated diffusion, now we have a molecule that is going to need some sort of assistance to get across the cell membrane. Might be a little bit larger in size, can't fit between the lipids on its own. And so now we're going to use those protein channels in order to get that movement to happen. So those protein channels, they span the entire width of the cell membrane, and they can be used to move products in or products out. So anything that would be too large to pass between those lipids without ripping them apart and destroying the cell membrane are now going to be funneled through that protein channel to make sure that we can still get it in and out of the cell, but we're not going to destroy the cell in the process. The next one that we have talks about osmosis. So if you look at that slide, osmosis is a term that a lot of people will use incorrectly. They will use diffusion and osmosis in place of each other, and you can't. Diffusion is talking about the movement of molecules. Osmosis is very, very specific to water. So anytime that you use the term osmosis, you have to be talking about the movement of water molecules, and that's it. If it's any other type of molecule, then you have to go to the word diffusion in order to describe it. So osmosis, same as what we've been talking about, requires no energy. Molecules are going to move where they are highly concentrated to where they're in low concentration. But as soon as you say osmosis, the only type of molecule that you can be talking about is water. And then the last one is filtration. Filtration, again, follows the same rules. We're not using energy, high to low concentration movement, but this is going to be used in places where you actually are going to have molecules moving through pores in order to get this done. And we talk a lot more about this when we get into our kidney chapter and we talk about the formation of urine. The process of filtration is very important to that process. So typically this is going to be used, and because the use of pores, that regulates, again, what moves in and out. The size of the pores are going to be adjusted to the size of the molecule that you want to move in or out of the area. So if it's a small molecule, those pores would be small, and make sure it's only that particular molecule that is able to move in and out. Anything that is on the larger size, so larger molecules, would be too big to fit through those pores. And so it just works as a process to regulate what is moving in and out of the area. The next one that we have talks about active transport. Active transport is a little bit different because now we are going to need energy in order to make this happen. And it is on a much smaller scale. There's a lot fewer places where we need to incorporate active transport. And again, that goes back to that energy requirement. Because it takes energy, we don't want to have to use this in a lot of different places because of the number of cells we have. It just would put too high of a demand on the number of calories we'd be able to put into our body in order to supply the energy to get this to happen. So active transport works a little bit different, using energy, but also the reason that it needs energy in order to happen is because we are pushing molecules in a way they don't want to go. Molecules want to go from where they're highly concentrated to where they're in low concentration. But when we talk about active transport, we're now taking molecules that are in an area of low concentration and we are putting them somewhere where they're already highly concentrated. They don't want to do that. If you think about if you go to a movie theater and you walk into the movie theater and you're picking out your seat, when you decide where you want to sit, you're not going to go to an area that's already jam packed full of people. What do? You usually find a place that has open seating and open area around it and maintain that kind of personal space. Molecules want to do the same thing. So they don't like to be packed in on top of each other, but in some places in the body, we need to see this happen. So, two of the best examples would be muscle contractions and nerve impulses. In order to get muscle contractions to happen, you have to have a lot of calcium ions on hand and in place. And so, active transport is going to take those calcium ions from places in the body and put them into these holding tanks in your muscle cells so that when you decide you need that muscle to contract, they're all on hand, they're ready to go, and that muscle contraction will go the way that it needs to. So that would be an example of active transport. We have to have a high concentration of these calcium ions in one location to make sure that when you need that muscle to move, it's able to do so. Likewise with nerve impulses. Nerve impulses also require ions in order to be able to fire. The two big ones are going to be sodium and potassium. And so again, stockpiling these and getting them all in one location so that when that nerve needs to fire, everything is already in place and the body's not having to try and gather those ions at that point to get your nerve to fire and go off. So those would be, like I said, the two best examples for why we would need active transport, where we would need to take different types of ions and pack them into one location so that whenever you need that impulse to happen, whenever you need that contraction to happen, they're already there, ready to go, just waiting. So with active transport, you also have a couple of kind of special items in there. You'll notice it talks about endocytosis and exocytosis. That is simply moving things in and out of the cell. But in the process, these items that are getting moved in and out of the cell are not just going to slip between the cell membrane. They're actually going to get packaged and they're going to get a layer wrapped around them when they're coming in or when they're leaving. And so as they pass through the cell membrane, they can actually take a part of the cell membrane and use that to kind of wrap around themselves to get that sort of packaging effect to happen. So endocytosis, the prefix endo talks about in, cyto means cell. So endocytosis indicates that something is moving into the cell. Exocytosis is going to, exo means to exit or to leave. Again, we see cyto, which means cell, so it's telling you that this item is leaving the cell. So in this case, the molecule would start inside and need to leave, and so it would simply get pushed out through the cell membrane, again, getting wrapped with a thin layer and packaged for its journey out of the cell. So endo and exocytosis simply indicate that you are moving something into the cell or you're moving something out of the cell. You'll also notice that it talks about pinocytosis and phagocytosis. These are going to be a little bit more specific. So endo and exo just tell you moving in or moving out. With the other two, it's telling you more specifically what type of molecule are we moving in or out. Pinocytosis, pino, P-I-N-O, is a prefix, means to drink. So it's telling you with pinocytosis that now we are moving some sort of liquid across the cell membrane. Phagocytosis, the prefix PHAGO means to eat. So now we've got some sort of solid material that we are moving in and out of the cell. So if you're just talking generally moving in and out, you can use exo or endocytosis. But if you know specifically you're talking about a liquid, you would use pinocytosis. And if you were talking about a solid, you could use phagocytosis. So the next slide that you guys have talks about hyper, hypo, and isotonic solutions. And this is really talking about the conditions that you have around the cell. And the most important thing to remember when we talk about hyper, hypo, and isotonic conditions is that we are talking about concentration. And concentration is different than equal number of molecules. And sometimes people get hung up on this because they want the number of molecules inside the cell to be the same as the number of molecules outside of the cell. That's not what we're talking about here. We are talking about the concentration of the liquid inside the cell being the same as the concentration of the liquid outside the cell. And that's a different concept. So it's not the molecules being equal. We have 3 inside the cell, so we have to have 3 molecules outside of the cell. It's not what we're going for. We want the concentration of the liquid to be the same inside as the concentration outside. So when we talk about hyper, hypo, and isotonic solutions, you have your cell and it has the cell membrane. So you have this closed environment of the cell itself. What we're talking about when we look at hyper, hypo, and isotonic solutions is we're talking about the conditions that are going to be right around the outside of the cell. So outside is the key thing to remember here. Hyper means that the conditions outside the cell are higher than the conditions inside the cell. So the concentration outside, the liquid outside of the cell, is at a higher concentration than the liquid inside the cell. The cell doesn't like this situation. It wants it to be at equilibrium. So the cell is going to do whatever it needs to do to get those two concentrations inside and outside to be the same. Well, we're not going to move molecules across the cell membrane to get that concentration to be balanced. What we're actually going to do is we're going to move liquid across the cell membrane to get the two concentrations to be balanced to each other. And so in the case of a hypertonic solution, that means your cell is in a concentration where the surroundings around the cell, higher concentration than the liquid inside the cell. What the cell is going to try and do is it's going to try and dilute the surroundings of the cell to get those concentrations to be balanced. Imagine that you make Kool-Aid and you put in way too much sugar and it's too sugary. One of the things that you could do is you could add extra water to the Kool-Aid to try and dilute it down and get the taste to where you want it to be. The cell is essentially going to do the same thing. It's going to take its own liquid contents and it's going to push it outside the cell and try and dilute that area around the cell to get the two concentrations to be equal. So what happens, if you think about that, if it takes its own liquid from inside and starts pushing it outside of the cell, what would happen to the cell size? No different than if you've ever filled a water balloon, you realize you overfilled it. So what do? You let some of the water out. The cell or the water balloon gets smaller. The cell will actually shrink in this situation. So what happens with that is the cell becomes smaller in size. You guys see this if you've ever seen a plant wilt. That is exactly what you see happening. The plant gave up its own water content to the surroundings around it. And so that wilting happens because those cells are shrinking down. The other situation is hypotonic. Hypo means below. And so now it's just the reverse. The concentration inside the cell is higher than the concentration outside of the cell. Again, to get those two concentrations to be equal, it's not about moving molecules, it's about moving liquid across the cell membrane to get the concentrations to be balanced with each other. So in this particular situation, what happens is now water from outside the cell is going to move into the cell to try and dilute the concentration in the cell to match outside. Again, if you think about water movement, now we're filling that water balloon with more water, so to speak. What happens to a water balloon as you put more water in it? Obviously, it expands. Now, what happens with the cell is it's going to reach equilibrium any way it can. And so it will continue to take on water until it reaches equilibrium and the concentration inside the cell is the same as the concentration outside the cell. If it can't do that, it will continue to take on water until it bursts. And so you could potentially have cell death with this situation because it will continue to take on that liquid until those two concentrations are the same, or the cell just can't take on any more liquid and it bursts or it ruptures. So in this particular case, a hypotonic solution, your cell is actually going to swell or increase in size. Isotonic, iso means the same. So this is telling you that the cell is in a situation where the concentration inside the cell is the same as the concentration outside of the cell. Now, there's still movement of water in and out of the cell. It's just happening at a rate that those two concentrations can stay in balance with each other. And so you're not going to see any change in the cell size. So hypertonic, the cell will shrink. Hypotonic, the cell will increase or swell. Isotonic, you will see no change in the size of your cell. So the next part of the packet gets into cell division. And we have two types of cell division that we'll talk about throughout the year. You have mitosis and you have meiosis. Now, right now, our focus is going to be on mitosis. And when we get into our reproductive chapters, then we will get into meiosis. Now, we have two different types of cell division because they accomplish two different things. And right now, we're just going to focus on mitosis, and we'll do a comparison when we add in meiosis with our reproductive section later on. So, mitosis is geared at number one, increasing the cell number. So for growth and development, you need to have more cells. Obviously, if you think of a baby at birth and then compare their body size at age 1, age 5, when they become a teenager into adulthood, obviously the body transitions and we increase in size. We're able to do that because of the process of mitosis. As we grow and develop, mitosis is going to provide the cells that we need for that growth to be able to happen. So increasing in cell size is going to be one of the goals of mitosis. The other goal of mitosis is going to be to replace dead or damaged cells. Now, when we're younger, we're constantly losing cells throughout our entire life. Now, younger ages, we are going to lose them pretty much at the same rate that we're able to replace them. So we don't notice any changes. We don't see wrinkles starting to appear. We don't see graying hair. We don't see some of the other things that we associate with aging because the death rate of the cells is equaled to the replacement rate of the cells. And that is because of mitosis. As fast as they're dying off, you have cells elsewhere that are reproducing, going through the process of cell division, and making sure that those numbers are in balance. As we age, unfortunately, that balance starts to get a little bit uneven and we start to have cells dying off at a faster rate than they can be replaced. And so that's why we start to see some of those structural damages or changes within our body. Now, with the process of cell division, it is something that is very regulated within the body. You don't want to have cells being produced unless you absolutely need them. And so this process of mitosis is something that is monitored and regulated so that it is able to make sure that the cells are needed and they have a place to go. There are different phases. The first one is prophase. The prefix pro indicates first. So prophase is the first part of mitosis. And you have a lot of things that are going on in prophase. Number one, the nuclear membrane is actually going to break down during prophase. And the important part of that is that when that nuclear membrane dissolves during prophase, is it releases the DNA to be able to migrate and move around the cell. Your DNA is usually very well maintained and protected in the nucleus so that it doesn't become damaged. But in the process of mitosis, we are going to go from one cell to two new cells. So cell division, we start with one, divide it, now we have two cells at the end of mitosis. Well, each one of those new cells needs to have a copy of everything. Needs to have a copy of all the DNA, needs to have a copy of all the organelles. And so making sure that that DNA is able to migrate and go into those two new cells is a very important part of the process. So that nuclear envelope has to dissolve or break down so that that DNA can be transferred into the two new cells that are going to come out of the process of mitosis. Now, the next part of mitosis is going to be the phase known as metaphase. Metaphase, the prefix meta indicates middle, and this is indicating where the chromosomes are actually going to be for a majority of this phase. So the chromosomes that are in the human cell are going to line up right in the middle of the cell. Now, the human body has 46 chromosomes in its cells, and each chromosome has a twin. So if you were to look at all of your chromosomes, a lot of times people will say 23 pairs. That's because each one of your chromosomes has a twin that it will match or identify with. The exception is the last set of chromosomes, and that is going to be your sex chromosomes. If you are a female, you will get an X and an X. They will match each other. But if you are a male, you get an X chromosome and a Y chromosome. So that would be the only place where you would not have that twin setup because you have the two different chromosomes, one coming from mom, one coming from dad. So the 46 chromosomes line up right across the middle of the cell. And this is a very important process. It has to be spot on in the middle. And when we talk about that, again, this is one of those places where you could just gloss over it and say they line up in the middle of the cell and leave it at that. But to get a full appreciation of what's going on here, what's eventually going to happen is there's these two structures on either end of the cell. So if you kind of equate the cell to the Earth, you have a North Pole and a South Pole, and you have the equator. So you have a structure that's sitting at the North Pole, you have another one that's sitting at the South Pole, and all of the chromosomes are lined up on the equator. Well, those two structures that are at the North and South Pole, they shoot out these protein rods that are called spindle fibers. And those spindle fibers will start to move towards the middle of the cell, and they will eventually attach to those chromosomes. Now, what happens is one from the North Pole will attach to the chromosome, and then you have another one from the South Pole that will attach to that same chromosome. And as soon as they attach, they don't wait for each other. As soon as they attach, they start a tug of war. And they will actually rip the chromosome in half. Half the chromosome gets retracted back to the North Pole, the other half gets pulled back to the South Pole. And that's how we get our genetic information into these two new cells that are forming. Now, because the spindle fibers don't wait for each other, if those chromosomes are not lined up right in the middle, if they were a little, let's say they were a little closer to the North Pole, well, then the spindle fiber from the North Pole is going to get to that chromosome first, and it's going to pull, and it's going to take the whole chromosome back to the North Pole. The South Pole does not get any genetic information from that chromosome. It's only by tearing it in half, half of the chromosome going to the north, half of it going to the south, that we get our genetic information dispersed between those two new cells. So the process of making sure that those chromosomes get lined up dead center is very, very important, because that is what is going to ensure that those spindle fibers hit that chromosome at the exact same time. That tug of war happens, it gets ripped in half, and each of those new cells that is forming is going to get the genetic information that it needs. From metaphase, the next one that we have is anaphase. Anaphase is known as the movement phase, and it's simply indicating that everybody's on the move. The chromosomes are migrating to the North and South Pole. All of the organelles that have been created to make sure that there's enough for two new cells are migrating to the North and South Pole, and everybody starts clearing out of the middle. So everything in the cell either goes to the North Pole or it goes to the South Pole. And the moving of those materials is done in a way to make sure that when the cell does divide, everything at the North Pole will take care of that new cell, and then everything that is at the South Pole will be enough to take care of that second cell. So anaphase, everybody's on the move, migrating to the poles, clearing out of the center to make sure that both of the new cells are going to have all of the materials they need to function. Which moves us into telophase. Telophase is going to be where the cell actually starts the process of dividing. And so the cell divides at the equator. So right where that equator is, it starts to pinch in, and it will continue to pinch in until the two sides meet, and then you'll have a clean break. And now you have your north cell, you have your south cell, all the organelles, all the DNA, because of how they migrated, and because the division happens through the center of the cell, everything should be in each of those new cells that is necessary for that cell to do all the things it needs to in order to live. Cytokinesis is the actual division of the cell into two new cells. So telophase is where it pinches in and continues that process, but once you have a clean break and you have two new cells, that is what we know as cytokinesis. Now, this, as I said, is a very regulated process, but unfortunately, there are some things that can break down and go wrong with the process. Cyclin is a protein that is present in your cells and is going to ensure that this happens like clockwork and that you don't have new cells being produced unless there is a need for them. Unfortunately, if that cyclin breaks down or something happens to it, now the process of mitosis will happen unregulated and out of control, and you will have cells being produced at a much faster rate. And on top of that, there's no place for them to go. So now you have this mass of cells that is being produced in the body, serve no purpose. There's too many of them, And when you talk about tumors, that's essentially what you're referring to, is that mass of cells that is produced that didn't serve a purpose. And it's all because that cyclin broke down and was not regulating mitosis in the way that it should. So you can have two types of cell masses. You could have a benign cell mass, which means that it is not, you have the mass there, but it's not invading, it's not taking over the surrounding tissues. Malignant is going to indicate that that bundle of cells is taking over, damaging the surrounding areas and surrounding tissues as well. So when we talk in those terms, obviously most of us are familiar with cancer, and unfortunately, probably all of us could talk on a situation in our own personal lives where we've had to deal with cancer in some way. And that's essentially what has happened. You had a mass of cells that were unregulated, producing at a higher rate than they needed to. And depending on if it's malignant or benign, would indicate how much damage it's doing to the body itself. The last one talks about cell death, which is a process known as apoptosis. And apoptosis is basically self-destruct. You have a series of cells in the body that will serve a purpose for a certain period of time, and then after that, we don't need them anymore. And we see a lot more of this in embryological development than any other place or time in our body. Two best examples, you have a cluster of cells that is actually going to fit like a mitten over your hand and your toes. And so during the initial development of the fingers and the toes, you have a lot of small parts and little pieces that are being fit together in order to form those fingers and toes. And so having that network of skin there to kind of help structure and anchor things while that development is happening is crucial to the process. But once they're formed, that skin is no longer needed. And so those cells are programmed to self-destruct at a certain point in embryological development. Okay, we've got the toes formed, we've got the fingers formed, we don't need that support anymore, so those cells will self-destruct and die, and you never even realize that they were there. Unfortunately, sometimes those cells don't get the message and they don't self-destruct, and so when you have people that have that skin, a lot of times it's referred to as webbing between their fingers and their toes, that's all that's happened, is that particular, those cells did not self-destruct the way that they were supposed to. But everybody has them at a certain time in that embryological development. Just some people, for most people, they destruct and go away. And in other people, it didn't happen the way that it was supposed to, and that skin layer still remains. The other place where we see that happen quite a bit is actually with the little flap of skin underneath your tongue. If you've ever stuck your tongue out, you'll notice that there is a thin band of skin that anchors the tongue to the bottom of your mouth. That in embryologic development actually comes all the way out to the tip of your tongue. And so it is much further out. And if you can imagine if that skin came all the way out to the tip of your tongue, how that would change your ability to move your tongue, you can see the benefit for those cells going through that process of apoptosis and clearing out to the point where they are now on your tongue. Again, for some people, it doesn't happen. Their cells don't get the message that they are supposed to self-destruct, and so they stay with them. And a lot of times that can be very easily corrected with just a simple surgery to go ahead and clear out that skin that didn't destruct the way that it was supposed to. So that is apoptosis. It's just referring to those cells that are programmed to self-destruct after they have served their purpose in the body and in the process of development. So that takes care of our cell chapter. Moving forward in Blackboard, go ahead and make sure that you also look for an assignment. There is going to be an assignment posted with this particular video. So watch it, take notes, make sure that you have those notes with you when you come to our next face-to-face meeting. And then in the announcement section with the assignment, I will also post the due date so that you guys know when you need to have that turned in to me. If you have any questions between now and our next face-to-face meeting, please give me an email and I will see you later.